Hello everyone. Welcome all of you. Warm welcome in Plant Science Pathshala lecture series. I am Sunita Jadhav and today we are going to study glycolysis. In glycolysis we will see what is glycolysis, its overview, investment phase, payoff phase and net gain. Glycolysis is an initial stage in the organisms that perform cellular respiration. What is glycolysis? We can break down word glycolysis as glyco plus lysis. Glyco means glucose and meaning of lysis is splitting or breakdown. Glycolysis is an universal pathway. It is common for aerobic and anaerobic organisms. Glycolysis does not require oxygen. It occurs in the cytosol of living cell because all enzymes required for this pathway are present in cytosol. First, we will have an overview about this process and then we will see various reactions involved in glycolysis. In glycolysis, one molecule of glucose, which is a 6-carbon compound, is broken down stepwisely into pyruvate, which is a 3-carbon compound. In this conversion, an energy is released that is captured in the form of NADH and ATP. So, glycolysis is energy releasing process. Glycolysis occurs in 10 steps, but for our convenience, we will study it in 2 phases. First phase of glycolysis is investment phase. It consumes energy. This phase completes in steps 1 to 5. And second phase is a payoff phase and it represents steps 6 to 9. It represents steps 6 to 10. Reactions which occurs in this phase produces energy. In first phase, glucose is converted into 2 molecules of glyceraldehyde 3-phosphate. This phase involves addition of phosphate, that is, it uses energy. In particular, in first phase, you need 2 ATP molecules to proceed reactions. Whereas in the second phase, glyceraldehyde 3-phosphate is converted into pyruvate. And it is in the second phase energy is actually produced. In particular, in second phase, you get 4 molecules of ATP and 2 molecules of NADH. Now, we will discuss first 5 reactions involved in phase 1. We begin with glucose molecule. First reaction is a conversion of glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is called hexokinase. Why this enzyme is known as hexokinase? We will see. The word hexo means 6, denoting the 6-carbon compound glucose, and kinases are a group of enzymes that catalyze phosphorylation reaction. So, the enzyme hexokinase transfers a molecule of phosphate from ATP to glucose and convert glucose to glucose 6-phosphate. In this reaction, ATP molecule is converted into ADP. Hence, this reaction utilizes 1 molecule of ATP. In the next step, you can observe reaction given here. Glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme isomerase. Fructose 6-phosphate is an isomer of glucose 6-phosphate. In the next, that is third reaction, fructose 6-phosphate is converted to fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase. In this step, there is addition of one phosphate group at the first carbon of fructose. Kinase enzyme transfers a group of phosphate from ATP to fructose 6-phosphate and ATP is converted into ADP. In next reaction, fructose 1,6-bisphosphate is split into two molecules of triose sugars, namely glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are 3-carbon containing compounds. An enzyme that catalyzes this reaction is called aldolase. Glyceraldehyde 3-phosphate, we say it also as a 3-phosphoglyceraldehyde. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers, so they are interconvertible. They are interconvertible in the presence of enzyme isomerase. These all reactions comes under the first phase of glycolysis. Energy is utilized in this phase. You can see first reaction and third reaction. These reactions actually consumes energy. Thus, in first reaction and third reaction, you observe that ATP molecule is converted into ADP. So, in first phase of glycolysis, you get two ATP molecules to proceed reactions. Now, you can see end products of phase 1 of glycolysis. Two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, are the end products of phase 1. Glyceraldehyde 3-phosphate can continue with the next steps of the pathway and dihydroxyacetone phosphate can be readily converted into glyceraldehyde 3-phosphate. Now let's look at second phase of glycolysis. It involves 5 reactions and it begins with glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. This reaction utilizes molecule of inorganic phosphate. Glyceraldehyde 3-phosphate oxidized to 1,3-bisphosphoglycerate with oxidizing agent NAD+. In presence of an enzyme glyceraldehyde 3-phosphate dehydrogenase. In this reaction, 2 electrons and 2 protons given out by glyceraldehyde 3-phosphate and reduce NAD to NADH2. In next reaction, 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate. This reaction is catalyzed by enzyme phosphoglycerate kinase. This enzyme transfers the phosphate group of 1,3-bisphosphoglycerate to ADP to form ATP molecule. It is clear that two molecules of ATPs are generated in this reaction from two molecules of 1,3-bisphosphoglycerate. Now we have two molecules of 3-phosphoglycerate. Let us see what happens with 3-phosphoglycerate. 3-phosphoglycerate is converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. This is simple rearrangement type of reaction, that is shifting of phosphate from 3-carbon position to 2-carbon position by isomerization. Further, 2-phosphoglycerate is converted to phosphoenol pyruvate. The enzyme of this reaction is enolase. Magnesium ions are also utilized in this reaction. Now come to the final, that is 10th step of glycolysis. In final step, phosphoenol pyruvate dephosphorylated to pyruvic acid in presence of an enzyme pyruvate kinase. The phosphate group at the phosphoenol pyruvate is transferred to adenosine diphosphate to form adenosine triphosphate. These all are the reactions that takes place in second phase of glycolysis. As the second phase begins with two molecules of glyceraldehyde 3-phosphate, it ends with two molecules of pyruvic acid. See carefully, out of 5 steps, that is reactions shown here, Energy is generated in three reactions. Reaction number 6. In this, when two molecules of glyceraldehyde 3-phosphate converted to two molecules of 1,3-bisphosphoglycerate, two molecules of NADH2 are released. See reaction number 7. In this conversion of 1,3-bisphosphoglycerate to the 3-phosphoglycerate, two ATP molecules are generated. Whereas in last, that is a 10th step, when two molecules of phosphoenol pyruvate converted into two molecules of pyruvate, two molecules of ATPs are also produced. Now come to the overall summary of glycolysis. Now we will see what is net gain of glycolysis. In the pathway of glycolysis, one glucose molecule is broken down stepwisely through 10 reactions, and then we get two molecules of pyruvate. Now let's discuss net reaction of glycolysis. You can see reactions begins with a molecule of glucose which reacts with two NAD molecules and two ADP units with two inorganic phosphate ions, and this is going to produce two molecules of 3-carbon pyruvate plus two molecules of NADH plus two ATP molecules. This net reaction states that one glucose molecule of 6 carbon gets splits into two molecules of 3 carbon compound pyruvate. There is utilization of two adenosine diphosphates since the 4 ADPs are used in second phase, but 2 ADPs are produced in first phase, thus there is a net use of only 2 ADPs. Thus, two molecules of pyruvate, two ATP units, and two NADH2 molecules are the net gain of glycolysis. In presence of oxygen, NADH can be reoxidized through electron transport chain and 3 ATPs form. As two NADH2 molecules produce during glycolysis, their reoxidation through electron transport chain generates 6 ATP molecules. So, net gain of glycolysis is 2 molecules of pyruvic acid and 8 molecules of ATP. I hope you learnt very well through this video. Friends, in next lecture series, we will discuss further steps of aerobic respiration like formation of acetyl coenzyme A, Krebs cycle, and electron transport chain. Thank you so much for watching video. Thank you.